Okay, so I will present what was the action of the work package 2 and work package 3, which are at the core of the user services. So that is what is producing the radioisotopes and what is distributing them to the users, or users coming to the work package 3 labs where they can do their own studies if they couldn't do it at home. So we have already seen the PRISMAP radionuclide portfolio, which has a lot of different isotopes. The red one, which are either positron emitters or electron capture decaying, the blue one, which are beta minus emitters, and the yellow ones, which are alpha emitters. So here's our chart of nuclides, where we find the stable isotopes in the middle. And then to the right, the blue ones, the more neutron-rich ones, and above, the neutron-deficient ones. And there's here water divide, because you always start usually from a stable target, which you have to bombard either with neutrons to go to the right side. And where do we find neutrons? In usually a high flux reactor. So remember, blue Cherenkov light gives the neutrons, not directly, but you can make a correlation. And this brings you to the right side where you have the beta minus emitters. If you want to go up, you have to bombard with charged particles, which you get from cyclotrons, and then you kick out one or several neutrons. So not all cyclotrons are painted in red, but when you open them, there's always some reddish copper electrons. So you can remember, red side, you are better off with a cyclotron. Now, every rule has an exception. There are some isotopes which are better made on the other side. For example, copper-67, which is clearly a beta minus emitter. There are very powerful methods to make those with cyclotrons. And in our work package 2 facility, DTU, they have produced copper-67 and shipped to users. And then to the top, that's a bit of merit, because there are some alpha emitters you can do with neutron irradiation, some with cyclotron irradiation, and some you can milk from some natural or other decay chains. And in our case, it was not a natural decay chain. It is a generator of thorium-229, which provides actinium-225, and that is based at GRC in Karlsruhe. So why do we need to get involved to supply radioisotopes for medical applications? Why don't you just order it? Now you can order things which are industrially produced. And if there's a market, industry will be happy to produce radionuclides for you. Technetium, iodine-131, fluorine-18, you don't need to make yourself, you can order. But that is only the case when the industry sees a big enough market to make money. And then there is no market yet, because you just want to do some mouse studies or some cell studies. Industry is not interested to make a big upfront investment into a new promising radionuclide, which maybe will not show up as promising. So that's a hen and egg situation, and this hen egg situation you can only break when you do an own effort, and this has to mobilize academic partners which have the capacity and capability to do what is needed. And here you see a summary of the different partners which are contributing to the work package 2. So first, you have to prepare a target to be irradiated. The first target is irradiated either with neutrons or protons or deuterons or alphas. So you have then an activated target, which you cannot use directly. You cannot inject activated gadolinium oxide powder into a mouse. It will not be very useful. You have to make physical separation or chemical separation or both. Then you have to label the product, make a characterization quality control, and you can ship it either to a user lab, or if the user doesn't have the capacity to handle such a new radionuclide, the user can come to a work package 3 facility. So to show it a bit more illustrative, that is like making the fundamental product, which in this case is the pig. But nobody eats pigs. You cannot eat pig. You have to transform the pig into a pork. before you can eat it. Now, this is not yet a form you can eat. You might need to make a physical separation. In this case, you separate the meat from the bones. And we don't eat pork carpaccio, so you have to make a chemical transformation to make it eatable. And then you can either take it like this, or you combine it with another tasty molecule, in this case here, some very rare, expensive stuff, to make a final product which is sterilized and used and can be shipped. So that is the principle in layman's terms. Now let's focus a bit on what those different facilities can help the users to exploit their experiments. So we have 5 work package-free facilities: DTU, Denmark, at CHUV, Lausanne, Switzerland, SCK, Mo in Belgium, TUM in Bavaria, and NCPG east of here. They propose certain services, and each of them has a special specificity. For example, if you want to have some exotic isotopes that don't have strong gamma lines, which you have to characterize very well, how much lanthanum-135 or erbium-165 you actually have, DTU is very well equipped with low energy spectroscopy. Then CHUV, they have microCAT, microspect, MRI, and since they are hospital-based, they are also very focused on translational studies, and you can even use clinical scanners. NURA is integrated into the campus of SCK. They can, for example, do longitudinal trials with mice to see therapy effects. Then you have TUM, which is again more similar to ANORA because they are also hospital-based. And finally, you have POLATOM, which has the nicest forest which we see here, and which we are going to visit on Thursday. So they have the capability to synthesize radio pharmaceuticals in GMP quality. Now, before you synthesize or use, you first must produce it. So let's now dive into the production, not in our pool. And the most used radionuclide in PRISMAP was terbium-161. It's about 30% of the deliveries, of all deliveries which were made, were terbium-161. And first you produce by irradiating gadolinium-160, captures a neutron, decays here. Now you have 2 chemically different species, which you have to separate. And this separation effort had been developed at PSI. You see here different radiolanthanides, which are separated. This is nicely isolated. On this plot, it looks very easy to separate. But you must remember that there is a lot of gadolinium and very little terbium. So it's that is exactly what was missing in the version. So it's like a ratio of 1 to 10,000. If you irradiate in a high flux reactor, if you go in a lower flux reactor, you have a ratio of 1 to 100,000. So that's like a needle in a haystack, and that requires quite some effort of a chemist to separate it. And since this, that's the second point which was missing. So you could imagine that you do it at home. So you have a friend who has some trigger reactor or some other reactor. You ask him to make the radiation. You look at the publication, how you have to do the separation. And then you hope, oh, let's try to do it ourselves. And that is the wrong conclusion, because you should not underestimate the effort to make such a chemical separation. So we had approached with colleagues at the other end of Grenoble, and I can go there by bike or by train, by tram. It takes 20 minutes. But eventually, it was better to make a wind test to PRISMAP, to make the chemical separation, because it would be an enormous effort to build up a chemical separation just for one quote. So for such long-lived isotopes, which live a week, it is much better to have a centralized production and dispatch from them. So what we did is that we irradiated and shipped to SCK or SCK irradiated. In fact, we have 900 kilometers to glide. But overall, it is better because with one produced batch, you not only serve one project, you ship something to Clermont and Bordeaux, you can ship something to Italy, you can ship something to US. So overall, you are winning when you have a long-lived isotope by a centralized production. And the more projects you have which can make use of the same isotope and you can synchronize it in the same week, the more efficient and sustainable is your production. Now, since PSI had already developed this, they have advanced it, and Zeynep has just shown. And then you can use the same to make a neutron-efficient isotope, here cyclotron produced. So here, the transmutation yield is lower, so you have to separate one product out of 10 to the 5 to 10 to the 6 target atoms. It's more challenging. But they manage, and they supplied it. You see the only thing which changes is now that this is called carbon on the above. And there's a complementary method where you irradiate at CERN, a tantalum target, you produce many different isotopes. Now it's no longer sufficient to make a chemical separation, which would give you all the isotopes of one element. You must make, in addition, a mass separation. And you do so with this mass separator here, which will define you isobars, which means all radionuclides which have the same mass. And separately, you will make some chemically selective ionization here, for example, laser ionization. Where it crosses, you get a monoisotopic beam. That is a quite universal method because you could, in principle, tune your mass separator to another mass, the laser to another wavelength, and you can do quite a lot of different wavelengths. So you see here this facility, and the advantages that you can even do it in a beam dump. So you don't take necessarily protons away from a main experiment. That's important to, like in a film in Hollywood, you always say no animals have been harmed, and at CERN it's important to say that no Higgs boson has been harmed by taking protons away. So why do you want to have 155 and 161? Because they belong to such a theranostic quartet. But terbium is not the only element where you have this. You see here there are some more. For example, scandium. And you have even an alternative which emits less far gamma rays, which might be better. Scandium-43 has been provided by PSI. And copper has been provided by DTU. And here we have a couple of lead-203 and lead-212. Where this is used for SPECT imaging, and this is used for alpha therapy. Now, this is not yellow, but it decays to daughters which emit alphas. So lead-212 was obtained from a radium generator where the radium had been produced at Milicis, while the lead-203, we can see, is produced by a reaction where you use deuterons, and when you emit 1, 2, 3, 4 neutrons, which means you need quite energetic deuterons, which are available at the Aronnax cyclotron in Nantes. So there was multiple batches of lead-203 which were produced in Nantes. And another thing which the cyclotron can do very well is the production of astatine-211, where you need alpha beams of around 29 megaelectron. And if you go to other isotopes, which will be presented tomorrow by the user projects, we have manganese-52, which is produced in PN reactions on chromium. And you see here a cyclotron in Riseux, which is being used for this at DTU. And also with PN reactions, they can produce palladium-103. So when you want to have such PN reaction where you just send a proton beam of less energy, which is sufficient to kick out one neutron, a smaller cyclotron does the job. And overall, with these different production methods which I have introduced, you can cover the entire radionuclide portfolio shown here. Thank you.